to everyone, Mr. Respected Chairman, my colleague speakers, distinguished delegates, and other participants. First, I would like to thank the organizers to invite me for this very important conference and the session. I am working at Indian Agricultural Research Institutes, and I have a vast experience working with sugarcane diseases. But my major specialization is on sugarcane viruses and phytoplasma. And today I am going to highlight very important disease of Asian countries caused by phytoplasma, that is sugarcane white leaf and sugarcane grassy shoot. Phytoplasma, just like in India, we have very other major diseases are fungal diseases, red rot, smut, poka boeing. But and not only for India, for all over Asian countries, fungal diseases are the major problem. But for the last decade, we have been observing that this phytoplasma disease is also becoming very, very important in several states in India and other other countries of Asia. So I am going to highlight phytoplasma. There are, you can see there are three major diseases, but two grassy shoot and white leaf, they are the same. Just by their in different name in Thailand, China, Vietnam, they are, because of the symptom, they are very much recognized as white leaf disease. But here in India is a very peculiar symptom, a grassy shoot, there is no tiller formation, just like a grassy shoot structure. That's why we have been recognized and called it as a grassy shoot disease. And beside this, we have another important disease, leaf yellows disease, which we know. The sugarcane, grassy shoot and white leaf, they belong to the same group of phytoplasma, and you can see the distribution on the basis of symptom. Most grassy shoot disease symptoms are recorded in India, Bangladesh, Nepal, Pakistan. However, the white leaf disease has been very, very common in the fields, sugarcane fields of Thailand, Sri Lanka, Vietnam, Myanmar, and other Asian countries. However, the sugarcane leaf yellow disease actually is a very complex disease caused by both by a virus and phytoplasma. Earlier, only in 1990s, sugarcane yellow has been discovered from Hawaiian countries in US. And then in other countries also, several scientists, they reported the occurrence of white leaf disease, yellow leaf disease. But in after 5-6 years later, scientists from South Africa, they observed that besides virus, phytoplasma is also associated in most of the sugarcane yellow leaf disease associated symptoms. So then we have reported this yellow leaf yellows disease in not only in Africa, but Cuba, Australia, Egypt, Brazil, and now we have been regularly reporting from other countries. So it is again becoming very important. And there is the Ramu-Eastern disease, first reported in Papua New Guinea, and it is very, first time it is very, very confused. Some some scientists reported virus, some scientists reported phytoplasma. So it is not only caused by a phytoplasma but virus, but virus is mostly found associated with the disease. You can see the symptom, very, very peculiar symptom, as I told you. You can see this grassy shoot-like structure in India. That's why it is given the name grassy shoot structure. However, in other countries of Asia, it is 100% chlorosis of leaves. That's why given the name white leaf disease. This is the distribution you can see. And very interesting, last decade, other than Asian countries, we have also observed occurrence of sugarcane white leaf disease in African continent. So it is again very important and challenging. Now we have to, and it is causing very severe losses. So that's now it is my request to pathologists and plant protection group to very, very careful for the introduction of this disease to other new countries. In India, you can see very wide circulation all over, from north, south, east, everywhere is a very, very important disease and causing losses up to 40 to 60%. So it has become one of the very, after fungal diseases, it is becoming very, very important in recent years. You can see the losses. In India, in ratoon crop especially, losses are much more as compared to the plant crop. And in most of the ratoon crops in tropical areas, subtropical areas and tropical, even Maharashtra, this is one of the major disease. And first time it is reported from Maharashtra, 1956. You can see Thailand, losses up to US 20 million have been recorded. Sri Lanka also losses are more than 20%. And Vietnam again in very recent years, it is very fast spreading to commercial varieties, and losses from 60 to 80% has been reported. There are several, we can diagnose the disease on the basis of symptoms, but it is not accurate, but because at early stage we never get symptom, only after 3-4 months, but sometimes even after germination, we can see the chlorosis symptoms. And so far, diagnosis is again very, very important for Phytoplasma diseases. Symptoms is again a very good criteria to identify the disease, but not 100%. We have to utilize molecular tools to 100% authentication for the identification of the disease. Then some staining techniques are also available. Electron microscopy is very, but it is not available everywhere and it is costly. But electron microscopy, we can cut the ultra-thin section and see the Phytoplasma bodies. Serology is there. Just a minute. So far, Phytoplasma is very, very peculiar. We are not yet culture Phytoplasma. That is the more important in the case of Phytoplasma. Although it is a bacterial member, but we can't culture this. Yeah. But PCR molecular based methods are quite authentic and very commonly being used for identification of Phytoplasma diseases on the basis of primers of different regions of genome of Phytoplasmas. And once we have sequence available, we can identify the group and subgroup level. Recently, we have developed LAMP assays for very quick and authentic identification of Phytoplasma. Symptomology, again I told, very clear symptoms you can see in the fields in different form. Closis of spindle leaves, excess still ring, proliferation of buds, and white leaf, and sometimes both, you can see both grassy shoot and leaf closis in a single affected clump. But as I told you, it is a very, very important disease of ratoon crops. You can see mostly more than 60% losses were recorded in most of the Asian countries. This is a picture of Thailand of Udon Thani, very severely affected Phytoplasma field with in sugarcane commercial variety in Udon Thani. There are some techniques available for identification by a simple compound microscope, by electron microscopy. I told you, once we have facility, we can cut ultra-thin section and observe very clear pleomorphic bodies of Phytoplasma in infected cell. Serological, because Phytoplasma, they don't have cell wall. So what scientists, they develop membrane proteins, this polycodon and monoclonal antibodies against membrane protein, and that is working very well. Again, but it is time taking, so it is not is recommended for number of this identification for number of samples. Only genome assisted tools are nowadays being utilized, and they are very well defined DNA protocols for extraction of Phytoplasma, either from the plants or from the insects, which are natural vectors of Phytoplasma. And there are already universal primers are available, which can, on basis of 16S ribosomal gene, and we can identify the Phytoplasma. It is very it is little tricky in the case of Phytoplasma, because titer is not, you can't judge the titer of Phytoplasma. So for that, if you have real-time PCR, that is the best for quantification of Phytoplasma, this infected cell. So real time for resistance also, we can utilize real-time PCR assay for the quantification of Phytoplasma, I mean grading resistance of different commercial varieties. Besides 16S rebel genome gene, now full genome sequence is available of several Phytoplasmas, and now we can also target different multi-local genes for the identification. And in the case of sugarcane, grassy shoot, we have already developed primers for different multi-local genes. You can see the list here, which can be utilized for very successful identification of Phytoplasma, either in the plant or in insects. And then for subgroup analysis, the USDA scientists, they have developed a very good computer simulated program, I5 classifier tool. Once we have sequence available for 16SR gene, that is a length of 12-15 base pair, you can utilize this tool for subgroup classification of Phytoplasma. Very, very easy, free, is available online. And on this basis, we analyze hundreds of samples showing a grassy shoot and white leaf, and we found they are very, very similar, 99 to 100% similar. Not much genetic diversity exists. That's why they are the both in Asia, whether it is sugarcane, grassy shoot or white leaf, they are caused by the single strain of Phytoplasma. Very recently, we have developed LAMP assay for identification. Here, there is no need of this PCR system, and simply room temperature with the help of different set of primers, we can identify the Phytoplasma. Is being utilized now. That is the challenge, I told you, still there is no successful culture of Phytoplasma. But there is a recent progress from Italian and Argentina and Mexico. They have developed some culture media. You can see the component, and they got little success of culture of Phytoplasma like bodies. You can see. But so far sugarcane, we are working. Once we get success, then it can be very utilized by grading resistance for sugarcane commercial cultivars. On the molecular tools, I told you we analyzed hundred of samples from different Asian countries, and we found again they are very, very similar, whether it is sugarcane, grassy shoot or white leaf. And yellow leaf disease, it is not belong to 11th group, it belongs to the first group, calcium Phytoplasma status group. As I told you, not much genetic diversity exists between sugarcane, grassy shoot and white leaf disease. Luckily, in very recent years, we got success in whole genome sequencing of sugarcane, white leaf and grassy shoot disease from India and China. So now we have more and more knowledge about genes involved in the genome, and in future we can utilize this information for resistance and identification purpose. Epidemiology is very important because they are causing very serious losses. So we must know the spread sources, natural spread sources in nature. And for this, we have recorded several insect species that are only leaf hopper and plant hopper. They are reported to as a natural vector for sugarcane Phytoplasma disease in Asia. And the maximum spread we recorded in the month of July, onwards of monsoon in India, and where average humidity is ranging between 25.7 to 22.9. So this again we are collecting all different environmental parameters to see the relationship for spreading incidence of Phytoplasma in different months, in different areas. Besides how it is transmitted in nature, sugarcane, is a vegetatively propagated crop. It is very well transmitted by set transmission, mechanical transmission through cutting, harvesting blades in different countries, and sap sucking insect, that is leaf hopper. I have listed here, you can see. We have recorded more than dozen leaf hopper species which are recorded to be natural vector of Phytoplasma. causing this white leaf and grasses. So in India, we have reported Dictyrocephalus vulgaris, Magis protecta, Cophna and unimaculata. Thailand is a major, no, Thailand sugar is again white leaf. If we talk about white leaf, it is very, very serious losses causing pathogen in Thailand. And two important leaf hopper species, Matsumira tetrix and Yamo tetrix has been reported. These are the real vectors which is very commonly available in most sugarcane commercial areas, cultivation areas, and these are the major problems for spreading from healthy field to infected field to healthy field. So a lot of studies has been done by our colleague Hanbu Singh from Thailand. In China, very recently they have reported two vectors, Tetragoniella and Clavia qualifana from Yunnan Sugarcane Institute, and it is being spread in, recorded in several these fields from Yunnan. Sri Lanka, again Miratetrix and Dictyrocephalus menai, another species of Dictyrocephalus has been recorded. Sri Lanka again is in Asia, number one we have India, Thailand and Sri Lanka. These are the three important countries where really this is a very, very important disease and very serious losses have been recorded every year. And Vietnam using Matsumira tetrix has been reported. And besides insects, weeds are also playing very important role in natural transmission of phytoplasma from sugarcane to weed and weed to sugarcane, vice versa. And in this connection, we have recorded two natural vectors from India, cannabis and Sphalanthus. These are list of species of vector. Already I told you, in India we have recorded more than 4 or 5 insects. However, 3 have been recorded as natural vector. From Thailand, these are the two major species, which are the very, and this is both are very common in all fields and causing playing a very important role as natural reservoir of phytoplasma. So for control, we have to think how to manage these insect vectors in field for minimize the incidence of phytoplasma. Then this is in Sri Lanka, Dictyrocephalus melanis. And to again a very interest, this sugarcane phytoplasma, this strain is very, very common to other phytoplasma strain reported from India, from not only from sugarcane, from coconut, erica nut, spear rot, oil palm, rice orange leaf disease, bamboo witch's broom, and other fruits, japota. So that is again very important epidemiology we have to see because their strain is more than 95% in future, they can be a pathogen of these crops also. In most cases, we have recorded mixed infection, which where losses are much more. We recorded sugarcane grasses suit with viruses, sugarcane grasses suit with fungi, fungal pathogens where losses are recorded much more. The second important pathogen is leaf hallow's phytoplasma. I told you, you can see the symptom. There is no very peculiar symptom yellowing of mid-leaf areas, but sometimes different patches also has been recorded. You can see irregular patches on leaf lamina in different commercial cultivars from India. And when we collected, we found either sugarcane leaf virus or sugarcane leaf hallow's phytoplasma, or sometimes mixed infection also. You can see variety of symptoms we have been observed. And my friend Dr. Viswanathan, he only concluded that this particular sugarcane leaf halo is again very, very important in deteriorating most of the commercial varieties. So again we have to look, that by epidemiological significance of this disease. Dr. Rao, please conclude within 2 minutes. Yeah. So already we have characterized sugarcane, we have also developed primers and we identified that it belongs to another group of phytoplasma. But in sugarcane leaf hallow's phytoplasma has been recorded from other countries also besides India and Asia. You can see Cuba, Australia, Mauritius, USA and Brazil, where another groups has been reported. So is again very, very important. Another vector has been recorded for vector Saccharomyces cerevisiae from Cuba, which belongs to first group of phytoplasma. Management is again very important. We have no, because vectors are involved, but no, it is not easy to control vector under open field condition, but we have to work on, already insecticides are being used, but the most applicable management approach is resistant, variety approach, and we are working on that. So far there is no grading scale for, now once we culture the this Phytoplasma, we can develop some protocol grading scale for successful, So some vector approach already we have been utilizing, but again, as far as environmental concern is there, we have to think for biological controlled approach. Several drones are being applied for wide application of insecticide in Thailand for management of leafhopper vectors. But hot water is very effective in India, and recently in Coimbatore, they have developed a very good system, is an STD, mechanized spraying of planting material of sugarcane, set treatment device, very, very handily, friendly, and very, very becoming very popular, not only for Phytoplasma, for other fungal and viral diseases. So it is already available now. You can go on SBI website and have detailed information of this treatment approach. Very, very applicable for farmers and factory for control of not only for Phytoplasma, for other important diseases. And they recorded,
Please.
And so other tissue culture approach has been used. So thank you so much.